Hello students, again we are with the same chapter. This is the last lecture for cell structure and organization. So today's topic of discussion is nucleus. Nucleus is a structure of a eukaryotic cell in which you can see that nucleus is distinct in a non-dividing cell, or we can see the same thing during interphase. In interphase, we can see that nucleus is made up of nuclear envelope, nucleoplasm, nucleolus, and chromatin network. One by one, if we discuss, nuclear membrane, which is called as a nuclear envelope, is double layered. It's a delimiting membrane of the nucleus, and it is separated from each other by a space called as a perinuclear space. Width of the space is 10 to 15 nanometer. If we see, the outer membrane is connected with the endoplasmic reticulum at places. It harbors ribosomes on it. While studying ribosome, we have come across that ribosomes are present on endoplasmic reticulum. But ribosomes are also present on the nuclear envelope, that is outer membrane of a nuclear envelope. The inner membrane of the nuclear envelope is lined by nuclear lamina. It is a network of a protein fibers which is going to maintain the shape of the nucleus. The two membrane along with the perinuclear space, it's going to help for separating nucleoplasm and cytoplasm of the cell. However, nuclear membrane is not continuous. It is having small openings at specific intervals called as a nucleopores. These nucleopores are guarded by pore complexes which are going to regulate the flow of a substance from nucleus to cytoplasm and vice versa. Here is a diagram. You can have a view of that which will give a detailed information about nucleus. Next. Nucleoplasm is nothing but the matrix which is present in the nucleus. As we all know, the matrix which is present in the cell, we are calling it as a cytoplasm. So similarly, the matrix which is present in the nucleus will be called as a nucleoplasm. But here there is one more name given to it. It is karyolymph. Karyo is nucleus and lymph is a fluid. So it can also be called as a karyolymph, which is containing various substances like nucleic acid, protein molecules, minerals, and salts. It contains chromatin network and nucleolus in it. Now, nucleolus is another component which is not bound by cell membrane. Nucleolus is made up of rRNA and ribosomal protein, and it is best known as a site of ribosomal biogenesis. That means nucleus is helping for the formation of ribosome. Now, depending upon the synthetic activity of the cell, there can be one or more nucleoli present in the nucleoplasm. It depends. For example, cell of a oocyte contain large nucleolus, whereas sperm cell contain a small inconspicuous nucleolus. These nucleolus are appearing as a dense spherical bodies present near chromatin network. If you want, you can review the last diagram in the previous slide. They produce rRNA and ribosomal protein, which we have already discussed, which are then transported to the cytoplasm and are assembled together to form a ribosome. So this is how the overall nucleus is going to help out. Next comes the chromatin material, which is also present inside the nucleus. Chromatin material is called as a genetic material. It is containing a genetic information in the form of a chromosome, but which are nothing but made up of DNA molecule which are associated with the proteins. As we all know, chromatin material, genetic material and DNA is one and the same thing. In a non-dividing cell, these chromosomes appear as a thread-like structure or a network which cannot be identified individually, but its network is called as a chromatin material. And this chromatin material is a DNA which is wound with the help of a histone and a non-histone proteins. As we all know that DNA is larger in size, which is accommodated in the small nucleus. So for accommodation, they are wound on these histones and non-histones. Now some regions which are present on the chromatin, some regions of the DNA are more genetically active called as a euchromatin, and some regions that contain more of the protein and less of the DNA are genetically inert, they are called as a heterochromatin. So these are two different regions which we see on the DNA. When the cell prepares to divide, that is maybe mitosis or meiosis, the chromosome coils, that means it undergoes condensation. At the metaphase stage, they become completely condensed and distinct, and we can clearly identify the chromosomes. You will study this process as well as the structure and type of a chromosome in another chapter in higher standards. Every species of a living organism has a specific number of a chromosome. Like for example, if we talk about humans, we have got 46 chromosome, that means 23 pairs of a chromosome. Now the nucleus is also having the entire genetic information, hence plays an important role in heredity and variation. It is a site for synthesis of DNA, RNA as well as ribosomes. It also plays an important role in protein synthesis. As we all know, in lower standard we have studied how the protein is synthesized from the information written on the DNA. The chromosome number being constant for the species, it is important in a phylogenetic study, and the nucleus is a master organelle of the cell. Lastly, we go to the cytoplasm, cytoskeleton. Cyto is a cell and a skeleton means a support system or a framework which is present inside the cell. Student, as we all know, we also have got a skeleton present in our body which is giving a shape and a support to our body. Similarly, cell is also having a skeleton which can be seen under light and electron microscope, and scientists have said it is a kind of a network made up of a fibril throughout the cytoplasm, specifically called as a cytoskeleton. It is made up of microtubules, microfilaments and intermediate filaments. Microtubules are made up of protein tubulin. Microfilaments are made up of actin. It's also a protein. And intermediate filaments are composed of fibrous protein. So these three different components is going to help for the formation of cytoskeleton. Cytoskeleton help in maintenance of the shape of the cell, contraction of the cell, mobility of the cell and cell organelles as well. Change in the shape of the cell and the cell division is also taking place with the help of cytoskeleton. Then we have last topic related to this chapter, that is centriole and centrosomes. We have a good diagram to view. Centrosome is usually found near the nucleus of an animal cell. What is this centrosome actually? These are nothing but the pair of cylindrical structure called as a centriole. We have been seeing during the diagram which are drawn for mitosis and meiosis, there is something which is star shape. This is just for your explanation. So, these centrioles are perpendicular to each other and are surrounded by an amorphous substance called as a pericentriolar material. Each cylinder of a centriole is made up of 9 cent of a triplet microtubules made up of tubulin. I think this, if we elaborate, you can see the centriole is having this triplet. Okay, and centriole is supposed to be having a base for the formation of the flagella and cilia as well. Evenly spaced triplets are connected to each other by the non-tubulin protein. At the proximal end of the centriole, there is a set of a tubule called as a hub. You can see inside this structure there is a presence of a connected triplets called as a hub. Okay, which is due to the means of radial spokes. Now, due to this proximal end of the centriole, it looks like a cartwheel that we can see, and the centrosome help in assembly of the spindle apparatus during cell division. It forms the basal body of cilia and flagella, which I have already discussed with you all. Thank you very much.